Hello everyone, welcome to BOC Sciences. Today, we will talk about PEG, a versatile conjugating ligand for drugs and drug delivery systems. In this video, we will review what PEG is, PEG synthesis strategies, PEGylation strategies, PEG quantitative analysis techniques, applications of PEGylated derivatives, and PEGylated products that are currently on the market. Polyethylene glycol, PEG, a compound composed of repeated ethylene glycol units, is also known as Macrogol. PEG and PEG derivatives are characterized as non-irritative, highly water-soluble and compatible, etc. At the same time, they possess excellent lubricity, moisture retention, and dispersibility that can be used as adhesives, anti-static agents, and softeners in applications such as cosmetics, pharmaceuticals, chemical fibers, rubber, plastics, papers, paints, electroplating, pesticides, as well as the metal processing and food processing industries. PEG conjugation is a rapidly evolving strategy to solve hurdles in therapeutic delivery and is being used as an add-on tool to the traditional drug delivery methods. Chemically, PEGylation is a term used to denote the modification of therapeutic molecules by conjugating with PEG. Meanwhile, efforts are constantly being made to develop novel strategies for the conjugation of PEG with these therapeutic molecules in order to advance the current applications. These strategies are specific to the therapeutic system used and also depend on the availability of activated PEGylating agents. Therefore, prior knowledge is essential in selecting the appropriate method for PEGylation. PEG can be synthesized via anionic polymerization of ethylene oxide and any hydroxyl initiators. Hydroxyl group could be from water, ethylene glycol, or any diols, and they can also be derived from epoxyethane by ring opening polymerization. Usually, the polymerization process produces a family of PEG molecules with a wide Gaussian distribution of molecular weights. However, because PEG bearing only two functional groups limits further derivatization with other residues or targeting ligands, commercial PEGs are available with different degrees of polymerization and activated functional groups. To date, researchers described a number of different approaches for PEGylation. The first generation of PEGylated products largely involve nonspecific and irreversible PEGylation of target proteins with linear PEG chains. Meanwhile, the demand for bioresponsive drug delivery systems led to the development of the second-generation PEGylated products, containing the covalently bound branched PEG chains at specific positions of the molecule, which are able to release drugs upon stimulation from the surrounding environment. In order to accomplish PEGylation, it is necessary to activate PEG moiety, which can be achieved by preparing a PEG derivative with a reactive functional group at one or both termini. Further, the use of the reactive functional group mainly depends on the substrate to be conjugated to PEG. As a result, PEGylation techniques can be classified into three components: PEGylation of proteins, PEGylation of small molecule drugs, and PEGylation of nanocarriers. Next, we will describe the significance of each PEGylation technique in detail. Because peptides or protein-based drugs induce strong immunogenicity and trigger the production of antibodies in the body, thus reducing the overall efficacy and eventually causing allergic reactions. Therefore, researchers seek to use soluble inert polymers, such as polyethylene glycol, PEG, combined with peptides or proteins to solve the dilemma, which can not only reduce the immunogenicity but also extend half-life while retaining its biological activity. PEGylation is a popular technology gradually developed in the late 1970s. At present, there are more than 10 PEGylated protein drugs on the market with excellent clinical medical effects. Peptide and protein PEGylation usually cover the following strategies: site-selective PEGylation, enzymatic PEGylation, as well as releasable and non-covalent PEGylation. Site-selective PEGylation mainly includes a nitrogen terminal PEGylation, which involves disulfide bridging using specific cross-functionalized monosulfone PEG react with protein disulfide bridge or PEGylation of proteins with unnatural amino acids and intine-fused protein, and both protein types are obtained by recombinant technology. Then, enzymes, due to their high specificity and selectivity, have also been considered tools for peptides and protein PEGylation. Transglutaminase-mediated PEGylation is the most commonly used method, which catalyzes the addition of acyl residues to primary amines. Moreover, glycol PEGylation, involving two enzymes to mimic oxygen glycosylation, aims to solve the limitation that threonine and serine in the case of oxygen glycosylation in asparagine cannot be PEGylated directly without affecting other traditional PEGylation sites. Furthermore, releasable and non-covalent PEGylation strives to overcome the loss of bioactivity resulting from PEG coupling at the receptor binding site. For the releasable PEGylation, tailor-made PEGylating agents containing hydrolyzable linkers are exploited to allow a slow and controlled protein release in vivo. As regards the non-covalent PEGylation, the polymer coupling does not involve a covalent bond, but instead, the polymer interacts with the protein through hydrophobic interactions or coordination bonds. Nanocarriers, such as micelles, liposomes, polymeric nanoparticles, and inorganic nanoparticles, are promising tools for controlled drug delivery or imaging in cancer therapy and many other applications. Up to date, a number of nanocarriers have been approved for clinical treatment of a variety of therapeutics. Liposome is an ideal drug delivery carrier. It possesses targeted properties, extended blood retention time, and higher organ distribution selectivity, which can improve the efficacy of drugs and reduce toxic side effects. Ordinary liposomes have the disadvantage of being easily cleared from the systemic circulation by liver and spleen macrophages. In this case, PEGylation can solve the above shortcomings, making liposomes stay in the blood for an extended period of time and enhancing the passive targeting function of drugs. Moreover, it is simple to prepare PG phospholipid derivatives in advance, which has become the current research focus of the PEGylation technique. Micelles are frequently the preferred choice for anti-cancer drug delivery. Preparation of PEGylated micelles mostly utilizes PEGylated polymers or lipids through synthetic approaches. Block copolymer micelles with PEG coronas have emerged as drug delivery systems with great potential, as they combine biocompatibility with the synthetic versatility of PEG. Furthermore, various activated PEGs can be used and exploited for these systems, such as block copolymer structures, providing control over the type and stability of covalent linkage formed. Biodegradable polymeric nanoparticles are advanced drug delivery systems due to their controlled release and targeting capabilities. However, after intravenous administration of polymeric nanoparticles, they are cleared by the endothelial reticulum system within a few seconds or minutes. Thus, in order to overcome this shortcoming, hydrophilic PEG is introduced to modify the polymer. The introduction of PEG will not only affect the biodegradation behavior of nanoparticles, but also affect the release and distribution of drugs in the body. Polymeric nanoparticles are usually PEGylated by physical surface coverage with PEG or PEG derivatives, thus, preparing nanoparticles with PEG block copolymers and grafting PEG onto the surfaces of nanoparticles. Small molecule drugs, especially anti-tumor drugs, are generally restricted by poor water solubility, short half-life, poor targeting of biological tissue distribution, and high toxicity, which greatly limit their clinical applications. PEGylated small molecule drugs can significantly improve the water solubility of drug molecules and in vivo pharmacokinetic parameters, as well as lower the related toxicity and side effects, which opens new development prospects for many small molecule drugs with significant curative effects but are limited in clinical applications. The most commonly used conjugating agent is linear monomethoxy polyethylene glycol, which couples with one end of the activated small molecule drugs. Although this strategy has few side effects, reaction conditions can be accomplished easily. Through such simple coupling reactions, the physical and chemical properties of the drugs can be modified, along with improved solubility. However, there are shortcomings of linear PEG that cannot be ignored, which include limited conjugation sites and low drug loading. These drawbacks can be overcome using various chemical modifications to obtain branched PEG, forked PEG, and multi-armed PEG. For example, forked PEG provides multiproximal reactive groups at the end of one or both ends of a linear PEG chain, enhancing drug loading by increasing active sites. Although several PEGylated therapeutics have entered clinical trials, the successful translation to clinical use requires an accurate assessment of pharmacokinetic and pharmacodynamic parameters in animals and patients. Sensitive in vivo quantification is important for evaluating the stability, metabolism, and bioavailability of PEGylated compounds. Strategies used for quantification include colorimetry, chromatography, radiolabeling, and biological and enzyme-linked immunosorbent assays. This table summarizes an overview of different analytical techniques used to quantify the PEG molecules and their applications. For example, enzyme-linked immunosorbent assay is a commonly used method for analyzing PEGylated protein concentrations in serum samples. As an amphiphilic polymer, PEG is soluble in water and most organic solvents and are characterized by good biocompatibility, non-toxicity, and low immunogenicity. It can be excreted through the kidneys, so no accumulation of PEG is observed in the body. In addition, PEG can easily bind with proteins, nanocarriers, and small molecule drugs after the terminal hydroxyl group is activated, thus being widely applied in various biomedical areas, such as drug delivery, imaging diagnosis, pharmaceutical preparation, and tissue engineering. For example, camptothecin is the progenitor of a series of derivatives actively against several tumors by inhibiting topoisomerase I. Pegamotocin is a prodrug obtained by coupling camptothecin to a diol PEG of 40 kilodaltons. This drug is linked through an ester bond involving a carbon-20 hydroxyl group and a carboxylic group of PEG. This approach aimed to extend the drug's half-life in blood by PEGylation and to stabilize the active lactone configuration of camptothecin by acylation. The value of PEGylated products in the pharmaceutical and biomedical industries is now well established. PEGylated bovine adenosine deaminase adagen, marketed by Enzon Pharmaceuticals, was the first U.S. FDA-approved PEGylated protein for the treatment of immunogenicity syndrome. A large number of protein and peptide pharmaceuticals have entered the market following adagen. Nevertheless, many PEGylated proteins and delivery systems are under clinical trials or at the developmental stages. Various PEGylated products which are marketed are listed in the tables. BOC Sciences is committed to supplying high-quality PEG derivatives, PEG copolymers, and PEG raw materials for academic research, institutional research, biotechnology companies, and pharmaceutical companies to meet the needs of the vast majority of customers. We also provide professional and cost-effective PEGylation, surface functionalization, PEGylated biodegradable polymer synthesis, monodisperse PEG synthesis, and PEGylation method verification services with fast turnaround time and high quality. If you are interested in our PEG products and custom PEGylation services, please contact us in advance.